So with that, I'd like to introduce our speaker for this evening. Dr. Shawn Wang is a professor of computer science at Cal State University Fullerton. He's going to tell you a fair bit about his experiences there. He holds degrees from universities in China and the US, as you can see here. And he has a variety of research interests, include data mining, big data, cloud computing, bioinformatics, and cybersecurity. And he's a member of the ACM SIGMOD, and the IEEE Computer Society. So please welcome Professor Shawn Wang.
I want to share what I learned when I was doing this research in here. And when I was an undergraduate student, I didn't take biology. That in China we do it there, and even high school I didn't take biology. They didn't have it. I take chemistry, I take physics, but I didn't take biology. In the university undergraduate degree, I was in math department, so I didn't take biology. So if I sound overexcited, please excuse me because this is the first time I learned so exciting stuff. And some of you may know a lot better than I did. And of course, you have a better ability. So as I said, the background of me is that I got into this area just when I was working on my Ph.D. thesis. And that is so exciting, I get into it. So my topic right now, I'm teaching mostly computational bioinformatics. And I wanted to share with you what I learned mostly in this area. Okay, so this is about me. I received full scholarship to pursue PhD degree in U.S. in 1995. And at the time when I came here, I have $500 in my pocket. That's all I have. So the university paid me, give me free tuition, everything, and pay me about $12,000 per year just for basic living. So I joined Cal State Fullerton in 2000, and now I'm currently a full professor. I received early promotion in 2005. And so I was department chair from 12 to 15, just try to contribute a little bit more to the department. And I helped establish the Center for Cybersecurity, and currently I'm serving as an assistant director for outreach. And these are the two areas I most focus on. One is bioinformatics, make life better. Another one is cybersecurity, and I advocate this anywhere I go, because in cybersecurity, we do need to have more people to work in that. This is an area it cannot wait. Bioinformatics, where we can do it, right? Next year, 10 years later, fine. Security, if we don't have people to work in that area, we're going to be in trouble because of all this going on there. So I was heavily involved in some of the international conference of the cybersecurity thing, and I was when I was in sabbatical leave, actually I worked as an editor, and edit this almost over like a 1,300 page two volume proceeding what is called the current trends in computer science and mechanical automation, and I learned a lot from there. So my goal in the whole every year is I want to learn. I just want to learn something new. So that big forum actually gave me a lot of chance to look at many different ways computer science can help. And I was a technical programming committee chair and keynote speaker for this conference that's in Hawaii, and it's a major topic to talk about it. So the other thing I want to share is a little bit about my research. So the one research I did since 1997 is 3D structure, pattern in 3D structure. So I developed this kind of called delta B plus tree structure, and it's based on a coordinate system. So this example here, we have a 3D structure, and we try to discover, the pattern inside of all the three structures. And the way we did is to somehow decompose the structure into so-called triplet. So 3 points at a time, and something like this. So we start with the structure here, and then we decompose the structure into 3 points at a time, and get the coordinate system to go with it. Okay. So the idea is that if we because 3D means 3 points is the smallest match you can find in 3D structure, right? Because the 3 points, the smallest you can set up a kind of a coordinate. So we just attach the coordinate to the 3 point here, and somehow decompose the substructure into a lot of these cells. And when you find a match, then you're going to basically, again, the coordinate system actually fixed with the 3 point. So when the 3 point rotated or move to match the structure, then the coordinate system go with it, right? That understandable? Basically, you have a coordinate system. Okay, now I have 3 points, and basically they are fixed. So when you rotate the 3 point to match anywhere, the coordinate system will go with it, right? So when you move it or you rotate it, the coordinate system will go with it. Now, what's interesting is now that if you find 2 match, then they may be combined together if the coordinate system happen to be aligned to each other. Make sense? Right? Because again, the coordinate system fixed with the 3 point. Okay, so now the 3 point have to rotate to match the structure. Now you have another 3 point match, and they turn out to have the same coordination aligned together. Then this actually 4 point match. So that's the basic idea of looking for a subtraction match in a three-dimensional space, and happen to be very efficient in the way we work on it. So this kind of algorithm, of course, can be best developed using the GPU because it's coordination, rotating and moving inside that things. So eventually we are looking for match in the 3D system, so that we'll try to extend the match to a bigger match. And I do have students working with me in those early years, and one of the students actually worked on this one of the project, and he was an undergraduate student and very good student. So he applied to NIH a scholarship and get into the interview. He said 7 people get into the final interview, and he's the only one who come from a university do not have a PhD degree program. All the other 6 people are just from top university. But eventually he got to hired by IBM and doing PhD part-time. So he graduated with PhD in UCLA while working for IBM. So he's still working for IBM, but he now is a PhD graduate. So amazing student. So these are the what we did in using Java visualization, and at the time we were trying to look at the 3D structure of the protein, and this actually Dan just mentioned to me about the AlphaFold. It was early year we are trying to look at how the protein sequence fold into a 3D structure, because for those of you who are aware of how the protein function, the protein interact with the environment for interface, right? So how the protein fold actually is very important to its function. If the protein doesn't well, the functional part is not exposed to the environment, is not going to work well. So at the time we are looking into that, say, okay, well, most of research are focusing on sequence, but sequence doesn't really have everything, right? Because protein eventually is a 3D structure, okay? So we are thinking that eventually everybody needs to go to the 3D structure to see how the protein function. That's our logic. But eventually, what happened was actually start with the University of Washington in Seattle. They developed a game initially for protein folding. So they because what happened was that we do not know how exactly it folded at the time. However, if you tell me, how this looked like, there are ways to validate it, meaning that because of the biological property or geometry property, we can verify is this the correct way, right? So I was actually attending one of the international conference when I was attending in Duke University, and there's a professor from North Carolina. She was working on validate the data that's submitted to protein data bank. I don't know if anyone will be aware of that. There's a so-called protein data bank. Basically, it allows people to upload whatever they discover of the protein 3D structure. So a lot of people, it's just open to everyone. So lots of people upload, say, oh, we just discovered a new 3D structure, they upload it. But this professor in North Carolina, actually, she worked with her student to develop a program to validate the data. And she actually detect lots of this doesn't even make sense because of the coordinate that they provided. Meaning that if you have an atom of this size and the center of the atom get too close, it's not going to happen, right? That's physics, okay? So she was able to detect a lot of these things. Some of the data submitted are really not right. And this actually come to what we have now. Initially, when the internet, when you're looking in the internet, I tell my students, probably 30% of the data of what's in the internet are inaccurate. Basically, you cannot just rely on that. But anyway, at the time, University of Washington developed this game. They crowdsourced it so that people can play that game, right? Say, okay, we have this, sequence like this, okay? So just come to tell us how it falls. And it's a game. And lots of people come to play. Eventually, some did get the correct answer. That's the early day of this 3D structure. But eventually, AlphaFold get the solution now. So what we learned from here is that the protein sequence have a very specific way to fold if it's normal or it's healthy. So in other words, there's not really, once you have the sequence, there's not really many different ways to get it folded. There's only one way. If it doesn't get folded that way, you may have a problem. So the mad cow disease, some of you know that, right? The mad cow disease from Canada, they found out actually it's because one of the protein didn't fold in the right way. And that become this. So that basically, because of that, then we are saying, okay, well, now we have, it's like there's a solution for this. Now, what we are working on the 3D structure, as that useful in this area anymore because there's a right answer on that. So eventually we move to another area about, again, protein structure, trying to use what we developed. So this is basically surface system and work in the protein docking. Basically, you have two proteins try to, talk to each other, right? So we can compare the surface structure and see, So now we are not looking into a single protein and say, okay, well, how it fold, because we have a solution for that already. So we are looking at, how the two proteins dock together, like interact with each other. And this is often very useful, especially in drug design. Drug design, right? Protein, the drug, small molecule normally bind into a kind of protein and try to make it become some kind of medication for people. So drug design use this a lot. And because of that, then we developed this concept called the alpha surface. And again, it's very common sense to understand that. Meaning, if you have a circle and you're rolling through the surface of protein, if your diameter is bigger, then the surface you cut out is less, right? So if you have a smaller circle, then you'll be able to get into deeper structure. And this is the way we try to find out the surface of the protein structure. So then when we have a small molecule of a drug or something, we try to put in there. we have to make sure this protein is capable of taking that. So there is some kind of application we are trying to look into. But 3D structure has always been my work. So these are some results from the algorithm we developed. So you can see here, this is one of protein we get from the proteins data bank. So initially, it's all those atoms there, and then when we try to run the discover a surface, then you're going to see different surface using different circle of diameters. So just imagine that you have a ball of certain size, and you try to roll like on the surface. Okay, and then you're going to see a different result from the surface point. And this is another interesting result. We run this algorithm using this model. I think we downloaded from Stanford University. They have this bunny basically with many points. Like, so we try to run the surface algorithm, and then we end up with a different surface point. And this is another based Java algorithm try to help us to visualize this. And the student I just mentioned, she worked on both of these algorithms, so it was amazing. Did a lot of this work. So this is another toolkit we developed. And then just recently, I started another project with some of my students, and it was a very interesting project because it was high school student involved. Interesting, right? We have a Choi High, right? Everybody know helpful is the Choi High. The Choi High students are required, before they graduate, they are required to do 115 hours of internship for free. So I have them every summer. I have students come to me. There's one student, a lady come to me. Actually, she will need to do it next year, but she wanted to start early. So she come to me 1 year before she need to do it. And she worked with me on some of the research. She really like it, very excited. Then she told her friend. Her friend is 1 year senior than her, and she already admitted to Caltech. Is a freshman in Caltech, and then they come together and work on this project. Basically, it's what's called a C4.5 decision tree algorithm. And just to give you a very quick introduction, the decision tree basically can look at all the different, symptoms or whatever medical experiment, whatever. So for patient, so have a lot of parameters, right? They come out for me, okay? And then the decision tree try to build a model where it can very accurately detect. For example, we work on the diabetes disease, diabetes thing, right? And say, okay, yes, this patient has maybe 80% or 90%. This person may have a diabetes, or yes or no, right? Something like that. Okay. So what happened with this C4.5 is based on very solid mathematical thing here. So it's called entropy. Whatever algorithm that has a very heavy math, that's the one I like most. Because although I transfer from mathematics to computer science, I still I love math a lot. Because math is a kind of a beautiful principle of nature, describe nature in a very concise way, and always work very well. So what happened is that this algorithm was capable to say that if you have 7 different things, you come out for this detect, blood pressure, all kind of different parameters you get out from this person. Then we are going to look at which one is most important for this diagnosis of this disease. Okay, so that algorithm is capable of saying which one is more important. And what we are trying to do here is try to again look at all these different parameters and say which one is most important, and using this algorithm. I do learn a bit about this, how the whole process works. Basically, what happened was that those cancer cells eventually they are able to come back to get friends. That's the problem. So just like virus, RNA virus, they do not have DNA inside. So the only way they can survive is get to the DNA of human and then try to bind to those things and somehow transcribe those RNA to get to there. So when this get to a point where this cancer cell is able to get more of the environment cell to work like them, that is when it becomes a problem. Okay. Yeah. So if the cancer cell itself is separate, isolated, it will be done very quickly with the immune system. But there are two things that the cancer cell become a problem. First of all, it evades your immune system, meaning that the immune system somehow didn't detect that in the early stage. And then eventually those cancer cells was capable of getting more other cell to join them. So that's how the virus work as well. So virus as RNA, they cannot survive them by themselves. That's why killing people is not the purpose. That's what's amazing about the virus. If the person died, the virus die with them, right? So they didn't want people to. They just want to get more, to infect. So that's a strategy. So when I was looking at this virus, I was thinking, okay, actually in some sense, the virus they are working together better than human. When humans try to work on this, they have different opinions. They don't work together very well. But the virus, they all work together in a way that just they all have the same purpose. They never fight each other. Oh, well, this is what is that. They just say, okay, well, our goal is to survive as a population, right? A race. Okay, so they are all going that way. So this is very interesting thing to learn. Yes.
Hi, Dr. Wang. So I'm intrigued by that alpha surface that you're building. So I wonder, because I'm not in the field, so I don't know, how do you build that? Is there an instrument when you get the protein? Is there an instrument that help you derive that surface? Or do you kind of build a 3D model of the protein, then you kind of derive the alpha surface?
We mostly just use the coordinate that are given to us. Just in the protein data bank, for example, they already accumulate a lot of existing data, and we use those data to calculate the cell structure. Yeah, so we don't do our own wet lab, just try to find the structure out, but we rely on the data that are available. And this is actually it's a very exciting area. There's one thing I found very interesting. Once I worked with some students and they try to do this, and they found in Russia, they also have a protein structure database. They only was able to get in there once. They get in there, access that, and get some data. Second time it was blocked. So that's a very interesting thing. They basically, the website blocked them when we come from United States to try to access. Yeah, the first time they get something, but the second time they went there, it was blocked immediately. So that gives us some idea. Basically, this kind of data structure, resources right now are still very important because people are trying to protect it. So, yeah. But in the United States, we do have lots of structure already out there. But as I said, the one professor in North Carolina, she cautioned people, say, okay, when you are using the coordinate data, be careful with that. Some of the data may not be completely accurate. Yeah.
Yeah. So I think the history of this is that there's a lot of protein coordinate data that was produced through different techniques, like basic techniques, biological techniques like X-ray crystallography was one in the early days, and a lot of algorithms have been used to try to physically model protein folding. And then more recently, AI has been used. Like you mentioned the AlphaFold algorithm. Google has basically been able to come up with predictions for pretty much every protein that the human body would and other organisms would actually generate and produce models and confidence levels for those. So there's a lot of data out there now.
Yeah, there's one process called protein crystallization. They basically make a crystal out of protein and measure the coordinate. Yeah, might take 1 week just to one of them. and the temperature, everything must be controlled very carefully. Yeah, so that's how they did the crystallization. Yeah.
Hi, I have a question regarding this slide. The first statement is select the attribute of the highest information gain.
Mhm.
How do we determine that specific attribute? Because that will impact how we go through the decision tree
That's right.
and what we'll eventually obtain.
Right.
And the other thing is, are there any autocorrecting algorithms that are also available if we selected the wrong or the suboptimal attribute?
Right. Yeah, this actually is something we are trying to find out. See, normally this was called supervised learning system work is that we use a data set, right? So many data here, and if we train the model using this data set, and later on, the new data coming happen to be matching the data we use very closely, you can have a very accurate result. However, oftentimes the new data coming is not that similar to what the data we are using. So this is what sometimes called overfitting, meaning that you are training, getting into too much detail of the new data. That's why we try to say, okay, well, we may have 7 attributes, but maybe one of them is not that important. If we use that attribute value in the training set, eventually it's not going to help us.
So would you try, say, whatever number of attributes that you have determined that is most likely based on knowledge of domain will be valuable, and then you test on one and say, okay, yes, it looks like it's okay, or you say, nah, it doesn't cut the mustard, let's try another one until we find out which is the best? Or how do you proceed in the selection?
The calculation we did, basically there's a called entropy and come from the, yeah, basically we try to calculate, and that gives you the idea how this. So what's happening is that you're going to calculate for the whole data set in the very beginning, okay? And then you're going to find one attribute that's most important, and then use the attribute to divide the data. And then you get to a second level. And then you have, suppose you initially have 7 attributes, one of them and the other 6 of them is going to be in the next level. And what's happened that is interesting is that when you get into a different branch or a second level, the next thing you find may not be the same attribute. That's what makes this pretty exciting. So which means the first level you are using blood pressure, for example, and then after you divide this, and then after that, okay, when the person has a very high blood pressure, the next attribute you're going to look at may be different from when the person has a very low blood pressure. That's something that's exciting to find. Well, that's common sense, right? Meaning that, you're going to find out, okay, for example, blood pressure, okay, normal blood pressure, too high, too low, and then you divide the data according to that. And then in the second level, then you're going to look at different things, the other attribute. And then it turned out that in the second level, with different group of people, the attribute you find out may not be the same. It makes sense? Understandable, right? Okay. And this actually algorithm also has been used in the like a credit card, approval. People say, okay, well, is this person good for credit card, and something like that. And the similar algorithm, basically, first thing we are looking at is this employee or not employee, right? And then second thing you're going to look at, okay, for those people who are employee, different thing look at. For not employee, there's another set of things. Yeah, so that's why this algorithm is very interesting. Yeah, this actually is one of the very classical algorithm already in there. And if Google it, you can find out and it was ranked top 10 algorithm in supervised learning. Yeah, and what in the one thing interesting again to me is like that formula is the most exciting because it has something to rely on, right? It's not simply just use the data and train it and train that. We do have a formula to say, okay, well, I can do some of the factorization.
Dr. Wang, this equation of attribute selection measure for information gain, do you think this equation could ever be translated into computer programming code for real-time invasive medical instrumentation?
This itself is programmable already. So with the data set, you can definitely do the calculation. Yeah, using this. Yeah.
Sure. Our,
Well, yes. I know. Yeah, but what happened is this. Most of the time, we separate the training time from the real-time detection. So the training time allowed to have a longer time, maybe several days, right? When you have a huge data set, you're going to spend like many days to train the model, right? But once you get to the detection, it should be very short. So normally a case get in there, you will probably come out within 1 minute, several seconds, it's already done. Yeah. So this is always the case. In the larger language model, the same thing. ChatGPT actually was trained for data before 2022, and they didn't add more data because it takes so much time to train the data set. Not easy to redo it again. It just takes too much time, yeah.
Okay, well, thank you, Dr. Wang. Appreciate that.